Let's talk about the ways that our immune system may respond to a gene therapy that uses a viral vector. Gene therapy aims to treat a genetic disease by delivering genetic material, such as a working gene, to target the cause of disease. Genes can be delivered by different types of vectors, and many are derived from viruses. Vectors help transport the therapeutic gene material to the target cell by making use of the shell of a virus, also known as the capsid. To make vectors safe to use, all of the viral genes are removed. Adeno-associated viruses, or an AAV for short, are commonly used vectors in gene therapy because they have proven to be safe, efficient, and effective at delivering therapeutic genetic material in the context of some conditions. Even though AAV vectors can be safe, they can still trigger an immune response and can lead to a variety of reactions. In AAV-based gene therapy, the body's immune system recognizes the capsid and sometimes the genetic material as something foreign and reacts to how it would with a flu virus or bacterial infection. An immune response can happen right after you receive gene therapy or later on. Different signs, symptoms, and response times will depend on how the cells within the immune system respond. The innate immune system is the body's first line of defense. It reacts quickly, sending out signals that call in immune cells for help. While the adaptive immune system, however, is the body's slower, long-term defense. It involves special immune cells that can affect how well the therapy works. T cells may find and destroy cells that received the treatment. B cells may react by making antibodies to block the therapy, also called neutralizing antibodies. Your body also creates memory cells, which means a faster and stronger reaction to re-exposure to AAV, limiting the potential for future doses. Before someone can receive gene therapy, the immune system is tested to help predict how it will respond. A key reason is that many people are exposed to AAV naturally without knowing it, and therefore produce antibodies. People who have developed immunity to AAV may not be eligible for gene therapy treatment using that vector type. Pre-existing neutralizing antibodies produced by immune cells like B cells can develop to prevent a therapy from working. This is because the immune system is ready to attack the vector before it can deliver the helpful genetic material. Testing for these antibodies may be done to determine if a person is eligible for a specific treatment. The amount of antibodies a person can have and still be eligible, also known as the antibody threshold, can vary for each therapy and the organ being targeted. The immune system's reaction to gene therapy varies based on the vector, dose, target organ, and delivery route. These factors will guide the medications used to manage the immune response. Medications may be given before and for several weeks after receiving the gene therapy. Long-term monitoring is needed to watch for potential late immune system effects, making follow-up appointments an important part of the clinical trial process. Researchers are exploring strategies to better prevent or manage immune responses, improve outcomes, and make more people eligible for gene therapies. To learn more about gene therapy and its unique risks, benefits, and challenges, visit patienteducation.asgct.org.